All right, we're 5 minutes past the hour, so I think it's a good moment to kick off. I hope everybody's doing well. I think a good place to start is with some festivities. As most of you know, it's the Iranian New Year, or otherwise known as Nowruz. So congratulations. Right, let's dive right into it. So it's really a great pleasure and an honor to introduce one of our very own, Dr. Leila. She's the head of data science team at PhazeRo. She holds a bachelor's degree and a master's in computer science and a PhD in data science and computational biology from Sharif University of Technology. Leila's research is kind of quite broad and deep. However, Leila's most recent research covered applied machine learning to enhance doctors' ability to predict cancer evolution over time. And Leila's most recent publication is entitled Conifer, which is clonal tree inference for tumor heterogeneity with single cell and bulk sequencing data, where Dr. Leila and her team provided a novel solution to solve for genetic heterogeneity of a cancer tumor that develops during clonal evolution, right? This is one of the reasons for cancer treatment failure, as it increases the probability of drug resistance. Although Leila is an academic at heart, she has been working in industry for over 10 years. And this is both at the kind of very high-paced startups like PhazeRo, but also at large enterprises. So she brings a wealth and depth of knowledge to the team. So today we'll be hearing from Leila about some of the data science techniques used to unravel the mysteries of DNA. As most of you know, DNA is essentially a recipe book that provides instructions for the production of living beings. Today's webinar will be an overview of key bioinformatics and data analysis concepts, which are essential to scientists' investigation or investigating rather the functions of genes. Before we get started, this is just a small matter of housekeeping. We're definitely encouraging engagement in our community and interaction. However, I would appreciate if you could kind of keep your questions to the end, and we have an allocated time for a Q&A session. So without further ado, it'd be my pleasure to hand over the conversation to Leila.
Thank you so much, Masood, for a really nice introduction. I should say congratulations, Happy New Year, Iranian New Year. Okay, let's start. Today, I will talk about human genetic data analysis methods and models. Most of you are familiar with the human genome, which is made of thousands of genes inside every cell of your body. You will probably also be aware that the human genome has been sequenced. But what does that actually mean? And how do you sequence someone's genome? Let's back up a bit. What is a genome? A genome is all the genes plus some extra that make up an organism. Genes are made of DNA, and DNA is made of long strands paired of A's, T's, C's, and G's. Your genome is the code that your cell uses to know how to behave. Cells interacting together make tissues, and tissues cooperating with each other make organs, and organs cooperating with each other make an organ system, and then organism. I mean you. So you are who you are in large part because of your genome. The first human genome was sequenced more than 12 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that made up your own personal genome, all in a matter of minutes and for less than the cost of the gift to your friend. Genome is made of about 3 billion pairs of these letters, and humans have about 20,000 genes. Genes are important because they contain the instructions cells need to make proteins, and proteins do most of the work in your body, building, maintaining things like your muscles and organs such as your heart. In some cases, and a change in a gene could lead to a change in the protein, and this could affect your body. For example, how well your heart functions. 99% of the letters in our DNA sequence are same across the globe, and only 1% is different between you and your friends. Even though it may seem small, this difference has a significant impact on many things, including how you look and your risk of disease. In other words, your genome contains all the DNA associated with the physical characteristics such as height, eye color, and skin color. And you may also find out some information about the risk of developing common diseases, which are caused by multiple environmental and lifestyle factors. For example, you may have a DNA variant that means you have an increased risk of disease like type 2 diabetes. Remember though that this disease is also influenced by what you eat and how much you exercise. Some variants in your genome can give clues to where your ancestor lived. For example, some variants are found mostly in Asia, and if you have this variant, then it's likely that you have distant ancestor who came from that part of the world. Whole genome sequencing is a technology that enables scientists to literally read the exact order of the sequence of all the letters that make up your complete set of DNA of your genome. In order to this, a blood sample is taken and the sample sent off to the library where a machine breaks the DNA, breaks the sample and reads the letters code. And researchers then compare your DNA sequence to a standard reference sequence. And they identify the variant or difference between the two sets of letters, then interpret the code. The process of whole genome sequencing can lead to many different types of personal information. Okay, now let's dig deeper and talk about how genome sequencing can help to treat an illness that is the leading cause of the deaths worldwide. I mean cancer, that affected nearly 10 million people by 2020, you know, the year of Corona. The problem in clinical treatment of cancer is that the drugs are extremely costly and the benefits of the treatment are relatively limited. Over the last 12 years, this is the result that we took at cancer drug approvals in the FDA. The median overall survival benefits was almost 2.1 months, and the cost of therapy is 10,000 a month, translating to almost millions of dollars per life year saved. But why is cancer so hard to treat? it was the question, and it was the time to a data scientist to take the lead of that. Around actually this question, why is cancer is so hard to treat, formed the framework of the first big experiment about 10 or so years ago. The tumor of a patient who has the kidney cancer is considered for this understanding. And as you can see, the size of the tumor is around 10 centimeters, and multiple biopsies from the primary tumor and the patient metastatic place have been taken, and the whole coding genome of that tumor and all the samples were sequenced. Around 22,000 genes and almost 300 million base pairs were sequenced in very deep details to understand whether all the biopsies give same results or whether they give very different results. The result of this research was so interesting. Actually, as you can see here, each of these red box shows the mutation, and the blue one shows there is no mutation. I didn't bring the name of each mutation because it's relevant to my talk, but if you see here, you can see in the primary tumor, there is some kind of mutation, which is, you can see this mutation in all 9 different biopsies data. And also, there is some mutations can only be found in the metastasis area. And also, like this mutation, and also there is some mutation that can only be found in the main and primary tumor. And there is and this just didn't mutated in the metastasis area. It was the first time that the people understood in deep and in the details which gene mutated during the cancer of this person. But why it's important? It's important because when a scientist design a drug for killing the cancer cell, this drug is sensitive to this kind of mutation. If the drug is sensitive to this mutation, just will be able to kill this kind of cells, not all of the cells that are in the cancer tumor. But if the scientist designs some drug can attack the majority of the cells, therefore the tumor cells and the metastasis area, all of them can be killed by this drug. Researchers and others in the field used a tree model to explain how tumors evolve. Multiple subclones exist in this patient's tumor, multiple cells that are independent from each other, as I saw here. And there is a common trunk, which is the mutation are same in the primary and the metastasis area, which is called here, for example, VHL. If we design a drug to kill the cells with this kind of mutation, all of the cancer cells in body of the human will be killed. And another actually understanding tumor evolution has many clinical applications. For example, like what we thought we can identify targets for treatment And also we can understand metastasis development of the cancer. And also we can understand the pattern of the evolution of the cancer. Okay, great. It seems we have a key that we are able to open all of the doors, but why still there is cancer in the world? The research was for 10 years ago, but still a lot of people are dying from cancer all over the world. This is because two reasons. The first of all is the genomic data must be interpreted. This interpretation may provide some information. The scientist is still in its infancy, although the technology is improving each year. There is still much to be learned in addition, interpreting a person genome sequencing. The person genome sequencing is very complex, and scientists are still working out things like how to use the information to estimate a person's disease. This is one reason reading and interpreting and translating it's not easy. And also another reason is the DNA sequencing. It's not much accurate that we expected. For example, as I said, the DNA will be cut to different reads, and we should read each of these part of the DNA. Sometimes one or two parts of DNA will be missed, and it may cause some inaccurate in the sequencing data. And actually, approximately 300,000 base pair are error in the DNA sequencing, which made a huge limitation on using DNA for inferencing and use it as a magic key for resolving all the problems. Okay, that's it. I want to say thank you for all of you for your time to listen to my presentation. Now it's time to have some questions.
Yeah, hi. Can I ask one question? Am I audible?
Sure.
Yeah, hi, Leila. Your presentation was nice. So actually, my question is related with this interpretation error. So as per my understanding, I mean, how to mitigate this interpretation error or bias related with this DNA sequence? You talked about this interpretation error. So how to mitigate this error?
I'm not sure I understand your question right, because you are cutting. How we deal with these errors?
Yes. To be
Okay.
precise, this is my question.
Yeah. It depends on the problem or the hand for dealing with these errors. Actually, for example, we have two different types of sequencing. One type of sequencing are bulk sequencing, which means a group of cells considered together, and the wall of the cells will be removed and the DNA will be give from the DNA. And it's about bulk of cells. Another type of sequencing that we have is single cell sequencing, which we just consider one cell, the DNA of one cell, but as the DNA of one cell, it's not enough for sequencing. This DNA should be add more. They repeat the number of DNAs by copying the DNAs. It depends how we deal with these errors. It depends in which approach we use for sequencing. But in generally, for modeling, usually a statistical model will be used for consider this kind of error. And sometimes, according to the problem, some errors can be ignored, or consider there is a possibility for this error, and this make the models for inferring that this is different from each other.
Yeah, okay, thank you. So actually, I have one more in line with this question. So if we are, if we are taking this interpretation error in our model, in ML model, for example, then will it affect the entire performance of the model, accuracy of the model?
If we, if we ignore this error,
I'm saying if we are ignoring this interpretation error in our model building. I'm talking about the machine learning model. If we are developing any machine learning or deep learning model based on this DNA sequence data, and
I'm not sure the problem is my side or.
No, I think the challenge is on your side of the guest, because I'm.
May I ask you to write your question in the chat?
Sorry, I'll write in the chat box. Okay.
Thank you so much.
All right. Well, while that question is being typed, any other questions?
Yes, I have a question more related to the issue of sequencing the human DNA. So as you mentioned, like the whole process initially took a long time, and you mentioned like hopefully in the future it will be shrunk down. Are the limitations in this case more on the scientific aspect, like how do we read it, or is it something that machine learning could improve? Like, is it something like chemicals and the biology and the science of that, or could machine learning be one step that would make reading the human genome a lot faster?
A great question. Actually, I would say currently the process of sequencing is so quickly. I guess around less than 24 hours, it's possible to read. The exome sequencing of the genome actually, or target gen sequencing. But about as I said in previous, people was work to find an approach to read the genome, and it was something that needed a lot of time. It takes around 20 years to understand how they will be able to read the genome. But currently, a lot of machines for reading the sequence, it's developed, and these machines read the DNA so faster than before. And there is the two-sided. One-sided is, but for example, if you see here, they cut the DNA, they will have a lot of DNA, they will read this DNA, and after that, they will assign these parts of DNA to the reference genome. The computational part of this work is this assignment, or it's better to say sequence alignment. It depends on how depth of this reads. It takes time, but the most time-consuming parts, for now, I can say I can say the device and the machine are this. Because, for example, for all of this letter, they should take a shot from the DNA, and they mark different of T, C, G with different color and take a screenshot of them. And after that, according to the color, realize where is A, where is T, and where is C, and so on in the genome. And this is the process that takes time, and the computational part of that, which is alignment and mapping, it's still time consuming, but I believe it's in the, I can't say for sure, but there are a lot of tools for that now. And it's in a cutting edge of the sequence alignment.
Great, thank you.
I believe the question is what will be effect on ML model if include interpretation error? Actually, it depends on what does the model want to do. If, for example, if we are about to have a model, something like that, to predict the evolution, and we have a problem with the main mutation, and we miss the mutation. We will design a drug to kill the cells, and we missed one important mutation, and the cells with this mutation will be alive, and it means we failed in our work. And therefore, it depends how affect this error can affect. It depends on the model. It depends on the goal. Yeah. I should say. I hope I answered your question.
Yeah, thank you so much.
You're welcome. We have another question from Rashid. What are applications of using dynamic programming for gene analysis? I don't get your question exactly. Dynamic programming, you mean for what goals? I can say if there is something that we can answer by dynamic programming, you can use it, but according to my understanding, the dynamic programming algorithms are the algorithms need, it's mostly the algorithms in N2 order, and this kind of algorithms shouldn't be useful for this huge amount of data. Yeah, I believe that this is my answer to this question. Thank you. I hope I answered your question, Mr. Rashid.
Hi, can you hear me?
Yes.
Oh, thank you so much for your nice presentation. And I have a simple question. I'm curious about the application of GAN-based models, generative adversarial networks, regarding mimicking the process of producing new genomes. Based on your presentation, I think we can use these types of models in order to generate new types of genomes, for example, new types of creatures or something like that. So I would highly appreciate it if you can explain this, my curiosity in detail.
Yeah, sure. Yeah, you're totally right. Again, it's really useful in this domain because, for example, let's back to here, we are talking about some kind of mutation that should be aligned to the first part and another mutation which are from the child of these three. But is there any order for this mutation? We just know that this mutation happened, this mutation happened, this mutation happened. There is no order. Therefore, using the algorithm like GAN can help us to reproduce the order of the mutation. But for example, it can be one application for that. I believe its paper published recently can be in this domain exactly that. I bring an example of that about the cancer evolution and for predicting the evolution tree. I believe if you start to write a paper in this domain using this algorithm and this model, it will be published really soon. Okay, we have another question from Ali Tubi. Thank you, Mr. Ali. Each cancer has its own personality. How can we utilize central dogma codons data gathering in target traffic? Exactly. The fact that you said it's really true. Each cancer has its own personality. It's totally true. And but please consider this fact that the cancer in different the body of the different people will be a start and end. And by considering the evolution of the cancer for different people, maybe we will be able to find some common pattern between different peoples. And therefore, it's doable that to realize some patterns, to realize something in common. In a massive case, if we will consider the pattern of evolution in massive case, it will be doable to find a pattern for some kind of cancer. If we find, yes, I should say yes, if we find, if we treat the source, if we find the source of this. But we will, for example, if we find something it's common between all the cancer cells, we will be able to teach the immune system that go and kill all the cells which has this mutation. We will be able to do that. But what's make it limited? As I said, the sequencing error and also the sequencing error and how we will be able to inference the mutation and how we are able to find the evolutionary pattern of this mutation. Which mutation happened first, which mutation happened second, and so on. Thank you. Any other question?
I have a question that may be rather basic, but again, since we're chasing our curiosities here, it sounds like one kind of the challenges here is obviously the size of the genome. You know, how big is the size of the entire genome from a data perspective?
Yeah, really great question. Actually, it was a challenge when we were working with the genome data. The size of them was a really challenging problem. The size of the whole genome sequencing, it's more than 1.5 terabytes. I mean, just talking about the raw data, but if you want to have some process of the data, Also, and decode some information which it's in the result of the machine. As I said, we have a machine here that produce this sequence for us, and for decode the result of this machine, this amount will be increased. Yeah, I should say around more than 1 terabyte data actually we have.
Thank you.
You're welcome. Yes, covered all questions.
I have one, also, I would say a basic question, but I'm curious, looking or let's high level now on the future. First of all, if you could just give me like a high level understanding of how far are we, I guess, from developing tooling for doctors where they can actually, use basically the results of this research that's being conducted. So are we there on some level, or like how far are we from that goal?
Yeah, really, actually, a really good question. I can say about the cancer. There is a lot of big lab all over the world that start to using these techniques, using the evolutionary tree of the cancer for treating cancer and target therapy, but they are limited. It's not something that we will be used all over the world. As I said, it's still expensive to have sequencing, and there is another issue for sequencing. For example, when they took this tumor after surgery and have different biopsy from that, a lot of cells will be go around the body of the people, and most of them will be missed in the analyzing of the cancer. And maybe this is another reason that it's this approach, it's not the final solution yet. But there is a huge lab in massively do and work and use the result of this evolutionary tree, but and also there is still a lot of way to improve the models which we have for inference, and also improve the devices for the sequencing. But as I said, the cancer is killing a lot of people all over the world. This is the domain that the scientists are working on that. Hopefully in near future, we will see something for attacking to this disease actually.
All right, really hope so. Great, thank you.
Yes, thank you.
Hello, I have a question. Thank you very much for your presentation. I guess we all have understood that how genome sequencing can be helpful and informative for designing customized drug for a cancerous person. So I'm curious to know if there is any possibility to to use that knowledge after sequencing a cancerous cell in the person to find some solution to prevent such an evolution, such a mutation in normal cells, at least for the people in the same family. Is there any room to use that knowledge for preventing purpose?
Yeah, actually, I like the idea of that. When I was talking about the evolutionary of the cancer, I just consider the people with same cancer. But yeah, the fact that you said it's totally true. We can see to the ancestor of a person, and also we consider if there is a parent of the person has a specific mutation. Most probably, it will be happen in the descent of the person. Therefore, I should say this is another part that it can be considered for this evolutionary tree and make it more accurate and more useful, considering the evolutionary path of your ancestor and descent, and see all of them together. This is another interesting topic. Yeah.
Thank you very much.
Right. I went back to my last slide to say thank you from PhazeRo team. Really appreciate for your time.
Wonderful. Thank you so much, Leila.
Thank you. Great presentation.
Thank you so much. I hope you all have a good day.
Wonderful. Have a good night. Thank you.
Thank you.
Thank you so much. Have a nice day.
Thank you so much. Thanks for joining and attending. Please attend our next webinar.